Or where did we disconnect? I just want to know the slide where we connected. Because I was talking about cancer when I noticed disconnected. So did we all get here? Huh? Or were we at news this?
I think you were saying something about neurotoxicity before
Oh,
it's connected.
okay. Okay. So
Copper, yes, copper. When they try to mix with
can you
copper.
imagine? Okay. So um, toxicants that can induce oxidative stress, and that okay, this work was done in our laboratory, and it was done with the MSc student. And then I was able to say that copper is one toxicant we get exposed to in our environment, present in our people get exposed to it in one way or the other. And then I was still to must first of all just do and what exactly are you interested in? You look at that. Another thing I said is that you must also look at what is in your laboratory or within you that you can make use of. So, example, don't do a research. For example, I used a confocal microscope when I was speaking. Now go and do something that involves a confocal microscope. It will be waste of effort because you'll not be able to get that result out. So look at the equipment you have in your laboratory or in your environment, or the equipment that come to my lab. I have it. You can do it. Do you get? Look at that when you're designing your own research in your own little way. And then I also say that you should not be scared to do what you want to do because of the fund, that you just need to know the right thing to do. There are a lot of opportunities for biomedical researchers. So you just need to know the right thing to do, the right application to make, the right grant to apply for, the right fellowship to apply for. For example, when I was being introduced, they said I had a twice DBT fellowship. That I did in India. Yes. So you just need to know the right fellowship to apply for, or what am I interested in? What is the right fellowship to apply for, and where can I do it? So with that, you'll be able to do that. So you don't need to get scared. You don't need to get agitated about it. So I said copper, we observed that oxidative stress was induced by copper, and I began to say that we are saying oxidative stress. It was because for that period, that was what we have available to us. But now our laboratory have actually moved further. We do genetic work. We have something to do Western blot and all of that under Dr. Bolaji. The laboratories have already moved forward, or it has moved further. So you don't get limited. However, you can start where you are. You can start where you are. So I went further to look at MPTT, and I said that this toxicant is something that induces Parkinson's disease. It induces Parkinson's disease. And one way is that it depletes the dopaminergic neurons. And that is one thing that happens to Parkinson's disease patients. The dopaminergic neurons are destroyed. And so we saw that, can you imagine this picture where all those, even from the least dose, it was already showing the effect. Can you see that the control is still very up there? But the survival of those that already had that disease was already down. So the survivor is very important, because if any disease occur, as it begins to affect the different mechanisms, it will lead to shortening of lifespan. It will lead to shortening of lifespan. And so you also need something to say, oh, assuming you saw something like this, what do I use to now prevent this shortening? And so we have things in our environment. So one of those things we used here was the resveratrol gotten from grapes. And then we saw that resveratrol can prolong lifespan. Look at control, the black line at this point, and then look at what resveratrol, how far it have extended the lifespan of these flies. And so by the time they brought both of them together, it was able to protect against or ameliorate or restore the effect that was done by the MPTT. And that is what is when they say somebody has a breaking research and all of that. These are some of the things that we observe to know that somebody had a breaking research. And before you look at it, they will just say, the person have gotten a Nobel Prize. It's starting from little things, little things. So little things matter. So you must observe what is happening, and you don't do it orally. You don't do it haphazardly. I am saying this because I'm speaking to students. I also was a student. Don't do things. Don't get into your laboratory and just do it quickly. You want to rush out. You want to do it haphazardly. You just want to. You think it is your supervisor you are favoring when you are doing it. No. One thing is that you are training yourself. You are being trained. You are learning the process of science, doing good research. You are learning it, so you must not do it just any. The precautions and all of those things must be taken care of. You must understand what you are doing, and you must be able to do it properly, and you must be able to report the way it should be reported. And so I went on to say that the Drosophila is also used in cancer research. Okay, it's also used in cancer research. It's also used in cancer research, and one toxicant we used was arsenic. And then I said that for arsenic, if you want to do cancer research, you must first of all say, what should I use? What carcinogen can I use? And then you begin to look at the carcinogen. And one of those carcinogens was arsenic. And then you must also begin to say, which pathway should I use? And I was saying that for this, it was very interesting to know that it was the same mechanism through which this carcinogen affects animals or rodents. It was the same mechanism it was using in Drosophila. The same mechanism, not different. So it becomes interesting. So you don't allow people to tell you, you're using fly. The result is not important. No, it is very, very, very important. Was the same mechanism. I'm not saying that all of them may actually be the same mechanism, but it is interesting when you see that the same mechanism is done, and you must be keen to observe. You must also be keen to say, okay, how do I know that it is a carcinogen or it is carcinogenic on this animal? So that leads to the part where you sometimes you begin to miss if you have those equipment in line to affected and all of that, so you can look at all of that. Interestingly, one of our other senior students here in the lab, which is Mrs. Judy, carcinogen and nitrosourea and benzoate pyrene. And then the first thing she did was that she kept observing changing. Interesting, if you are using wider animal, you may not quickly see the phenotypic changes. It may take some time, of course. It may take a long time, but for flies, it may not observing the phenotypic changes under the microscope, and yet your observation of these two carcinogens just change the phenotypic changes in the eye of the fly. But that does not mean that it is eye cancer it causes. So I must say that. But the change, looking at the fly, and that made her to go further to do what she wanted to use it for. That flies, that carcinogen basically is used for ovaries, and so she checked the ovaries and she saw the disruptions and all of that. So you can do a lot of work, but as I encourage you, you must read. These flies can be used to do a lot of things. Another thing is that the flies also have genetic models, and so you can cross them. They are drivers that by the time you cross them, the progeny will carry that behavior, that thing. I must be able to identify, what will be the change. So it needs a lot of reading to understand this. What would have happened? By the time you cross them, it will carry, the progeny will carry, but it's not all the progeny, because you remember that when we did genetics, you cross. Some we have some things and all of that. So some it is a specific one that we carry it. So you might be able to select that specific one at and then be able to carry out your research. And then it was interesting to know that you know that Drosophila is used in personalized therapy for cancer. In personalized therapy. And so how do you do this? You get the fly, the gene of a human, somebody that is cancerous, get the gene. Assuming you want to treat the person, get the gene of that person, and carry out the gene expression to know the genes that are mutated, and then you induce that gene or you formulate a fly that has that mutated gene, the same type for that this thing, for that fly, for that human. And then you begin to treat with different doses and a different drug, different types of drug, different combinations of drug. And then with that, you'll be able to select a particular drug that will be protective in the fly. And then if you take it to the human, use that same drug. When they say personalized therapy, you're trying to treat a person based on his genetic makeup, his own genetic makeup, his own what his genetics is saying. Not just like no everybody just go to the clinic and say I have malaria, then you just give everybody's paracetamol or something. No, based on that person's genetic makeup, based on what the person is saying, there may be little variations. That is why you need that personalized therapy. And so you take it to the patient, and it was interesting to see that that same drug that improved the fly, it also improved the patient. It prolonged the patient for that, but after some time, because cancer is something that have a lot of phases, it can metastasize. And because that patient was already in the metastasis place, the thing resurface again, but it's actually prolonged the lifespan of the patient longer than what it was. So it resurface again, and the person was already tired, and then he couldn't do it. So this was some papers on personalized therapy where they use this drug, temetribine, and this other drug in treating a colorectal patient, and it was very good also for the colorectal patient. They had different combinations. This was the fly expressing the gene. This is a normal fly, and by the time they treated with different drugs, this drug was the best, different drugs. And so they took this drug to that patient. The person improved drastically better. So these are some things you can do with flies in biomedical research. These are some things you can do with flies in biomedical. In short, apart from all of those ones I've mentioned, neuro, cancer, diabetes, there's still a lot. Just Google. Don't just assume that it cannot be done. Google. Go to PubMed. Put this and Drosophila. You will see papers coming out. You just read about it, and then ideas will come. And don't forget to ask God for direction, for wisdom, because it is God who gives wisdom and also gives us strength to perform research also. So conclusively, Drosophila melanogaster is an essential model for scientific discovery and in biomedical research. Is an essential model. I know Nigeria is still taking it in gradually, but I think we have gone far. But it's an essential model. All other world, they've gone far with this. So it's a very essential model for scientific discovery. And it helps us to know the state of diseases. And then how can we also treat it? Although we still have a lot of questions yet to be answered with this model, but we must keep pushing, because through this model, a lot of questions can still be answered. So we have some books you can read here. We have this fly push. Just Google fly push. You can get that book, read a lot of journals on Drosophila, and then we have this fly-based database. You can get a lot of information from this website, the fly-based, and then I really want research body, the Drosophila Research Center, was opened by my Amos Abolaji, really been an interesting one for me because through that body, I've been able to reach out to so many lives. I want to thank him for that body which he opened. I also want to thank the Biochemical Society for the different grants they have given to me to host some Drosophila research trainings. I want to thank them for that. I also want to thank my institutions, Queensland University, where I lecture presently, for the for giving me this lecturing position to reach out to much. That was where I met my mentors. Thank them for that privilege, and then I must acknowledge my mentors. This is my so they are both my supervisors in my PhD. I want to thank my supervisor, Professor Yunke Odwola, for always being there. She has always been a mother. She has always been there for me. The mentorship, you don't go away from being mentored. If you don't come under somebody under an authority, then you may not go far. So you must keep being under somebody, like learn somebody. You must be able to submit to someone. Don't be someone that cannot submit to someone. So you must be able to submit to someone to learn. So I want to thank her for that leadership position. I also want to thank Dr. Amos Abolaji for always being there as a father. Thank him for we look up high to him. I want to thank him for the support. And I just sent him the link not quite long to join, and he did join within a short notice. Though I already told him ahead about this meeting, about the invitation. So I want to thank you all for inviting me for this presentation. Thank you very much. And thank you for listening.
Thank you so much, ma.
Thank you so much, ma'am.
Wow. Thank you so much, man. It's really been an awesome time learning from you, man. It's really awesome. In fact, I've learned a lot. I mean, a lot. Personally, I've learned a lot from the research process that you started from down to the explanation on Drosophila and everything. In fact, it's really awesome. Like I've been able to learn a lot, despite the fact that I'm not even a biochemist. I'm not in the field of life science, but the inquisitive of trying to know to learn more on life science and related and stuff have been like you even prompted me to go learn like you inspired your teaching inspired me to go learn more on Drosophila because initially actually I don't even know that we even use all these smaller organisms in research processes. I only thought of all these albino rats and some other organisms, all these bigger some other bigger organisms getting to know about Drosophila, which is a fruit plant and I mean a fruit insect and some other getting to realize that we could literally use some other microscopic organisms as well to in terms of research and they even have probably higher percentage of genetic modification with respect to human. In fact, uh, like so surprising to me actually. Like, but notwithstanding, I'll get to learn more about that. And at the same time, I really appreciate you. We at SCIENCOON, we appreciate the fact that you all know our invitation. In fact, it's a privilege to learn from you, man. We really appreciate you. And at the same time, we look forward to hearing from you. In case we invite you over again to come teach us, I believe we definitely honor our invitation once again. Thank you so much, man. It's really a privilege to learn from you. Man. Thank you so much. You really taught us a lot. Personally, you taught me a lot. Personally. I'm very, very sure of that. It taught me a lot, man. Thank you so much, man.
You're welcome.
So, so yes, thank you so much, man. So to us all, any question, please let's drop our question. Let's let's signify if you have any question for doctor, please. Any comment, any question, any observation, whatsoever it is you wanted to pass across, please. Let's the mic is over to us, please.
Let's ask questions. I must say that for you to be a good scientist or a good researcher, then you must be inquisitive. So the ability to ask questions can open a whole lot of doors for you. I know in our Nigeria system, we tend to keep quiet. Or maybe our lecturers have taught us, oh, don't say something in the crowd. Don't do this. Or from our family background. The ability to ask questions can open a whole lot of doors for you all over. Ability, like people get to know you as you ask questions. People get to see your level of intelligence and begin to say, oh, this person would be good in this. So let's not keep quiet. I encourage you to ask questions in case you have any questions. I think Alexander has a hand raised up.
Yes. So over to you, Alexandra, please. Ask a question, man. Okay,
Shegun,
Mr. Shegun, please over to you, sir. Ask a question.
All right. Good evening, everyone. Okay, I believe you can all hear me. Good evening.
evening.
Uh, okay, my name is Olabuyede Shegun, and I am the president of SCIENCOON. Okay, I will like to appreciate Dr. Agogo Ibo for honoring our invitation. And I also believe that we've all learned something new today, respect to Drosophila melanogaster in biomedical research. Um, okay, I want to talk about my experience at Drosophila Research Training Center at Bashoru in Ibadan. I was privileged to be selected among the 20 students here that were selected last year to be a part of the training on how to make use of Drosophila in biomedical research. Of course, Dr. Ogoyinbo, she's not a stranger to me. I'm already used to her. I know she's very sound, she's very amazing. So I'm really glad about this webinar. Of course, there we were actually taught how to undo Drosophila, how to rear it. No, I won't say rear, now how to culture it. Just like the way they always rear this albino rat that we use for research. So there's a way they actually like culture them, the preparation of their food, and also inducing certain disease if you want to carry out anything that has to do with research. So we were taught all of this. I remember my group, we induced Parkinson's disease using retinol. I remember she mentioned retinol today. So we actually did all of this. So it's a beautiful experience. If you are privileged to go to DRTC, that's the Drosophila Research Training Center. If you are able to go there, you will see a lot of things like one that's taking place there. And also, I remember they took us to University of Ibadan. There's a Drosophila lab there. For those that are very curious, you can you can go there. I don't know whether they allow people to visit the lab. I don't know. I'm actually thinking, though I've not discussed this with Dr. Agogo Yibo and Dr. Amos, the members at Cyclone here, maybe it is possible for us to probably come over for a form of excursion so that we can have access to the lab and also to introduce some of our members to how things work over there. Because I'm sure most of us here we are just hearing about Drosophila for the first time. I'm not just hearing about it for the first time, but some people they're just hearing about it for the first time. So it will be a very nice thing for them to go over there and then sit in themselves.
That is a very good one. I know we are open. We are open for you to come in one day. School visit, in church, secondary schools come in one day. They send their secondary school for their students for time out of school one day, and all of that. Um, so the members of SCIENCOON can come into DRTC for an excursion, one day excursion to see how it is used. Where I'll just speak with our team lead, and then it gives the go ahead, and then you also will tell us. By the time I speak with him, he gives the go ahead. You also interact with him to get the specific time that will be more feasible for DRTC to welcome you guys properly and all of that. But of course, we are all open-handed to welcome you to help you understand more about Drosophila.
And again, other question, ma, this is a question now. Is it possible for maybe a student in theory level going for Cyrus now, I mean industrial training, to actually like go over there for a form of training? Yes.
Yes, we
Yes, we
accept
accept
Cyrus
Cyrus
students to
students
study.
to study.
The person has to make an application through the Drosophila Research and Training Center, and the team will also get in touch with the person. But we have Cyrus students, those doing Cyrus in GRTC. I think since 2 3 years, we have always been having Cyrus students coming in to do Cyrus in GRTC. So it's possible for you to come into GRTC and to just lab UI to do Cyrus. And of course, you are going to have a good experience when you come here to do a Cyrus IT. So that is it. Yeah, Solomon, you can go ahead and ask your question.
So I guess Mr. Shegun is done with his
Okay, good evening, ma.
questioning. Mr. Solomon, we can see you, please. Over to you, sir.
Dr. Solomon is Dr. Solomon. He's a lecturer here in Futa.
Hi, sir. Well done, sir. Welcome,
Oh,
sir.
yeah, good evening, ma. Good evening, everybody. I'm happy to be here, and I was so thrilled with the
Welcome,
lecture.
sir. I'm sorry for the welcome, sir.
I don't do research, but I am interested in biochemistry because I'm in soil science and do soil biochemistry. And when I saw the advert, what do I call it now? The a call for this
Call for this
webinar on
apply.
the
Webinar on the
Femi status. I mean Shegun status.
status.
I said
I
that
said
knowledge, and I like to acquire knowledge from every area. And since it's a Sunday evening that I'll be a little free, let me be here. But being here has been tremendously wonderful to me because the lecture was fantastic. I want to appreciate our guest lecturer. The only area that caught my attention, no, everything caught my attention there, but the one I'm interested in here is because I do research on uh, some plant materials that we can use to uh, as additive to fertilizers to actually improve soil and environment health. And the areas we are working on at the moment, we are gathering candidate plant materials and extract that we inhibit certain enzymes in soil that enzymes that will produce glucose, because glucose, as you know, is a precursor to uh, carbon mineralization, which will eventually release CO2. The things I was hearing people saying about the bitter leaf and bitter cola, for this uh, that we use to as additive to improve the health of diabetic patients. Because we have realized that some of the enzymes that will eventually produce sugar and release into the blood, they are also uh, applicable in the soil, like the glucosidases. So we have done some research on some plants that the extracts and exudates are able to inhibit beta glucosidases in soil. And if not here, maybe personally I can approach you to give us that some candidate plants, medicinal plants that have been used for to manage diabetics. They will be helpful to us or applicable to inhibiting some enzymes that will aid carbon mineralization, so that we can reduce carbon mineralization and improve carbon sequestration. I don't know if you're familiar with what I'm saying, but uh, maybe I can explain better when I talk to you. Thank you,
It's
ma.
okay. Thank you so much, Doc. I'm very grateful. Thank you for joining us on this webinar. I'm very grateful. It is nice to know that a doctor lecturer also joins this webinar. So I'm very grateful for that. Of course, we can link up, sir,
Yes.
so that we can get some plants. I just think what we need to do is to go on literature search to know on plants, most especially in Nigeria, that is used in reducing the blood glucose level. Because the fact is that the herbs and the plants are there for us for our benefits. But it just depends on the dose or how much you take them that the benefit comes. So, and of course, I know bitter leaf is one good plant that reduces blood glucose. If there is diabetes, it reduces blood glucose drastically. And bitter cola is also another very good plant. There is this king of bitters also. There's this king of bitters also. It's also very good. People also use aloe vera, make it something. So there are a lot of papers that we do on this. So let's just we can link up and then we'll just look at that together, sir.
Thank you very much. I really appreciate it.
Okay, sir. Thank you, sir.
Well, thank you so much, ma, for your time. Any other question, please? Let's keep it coming. Any other question? Any other question, please? Oh, since I guess we've all been ruminating on what doctor has taught us today, and at the same time, with the intention of going to make research and learn more about Drosophila and some other related knowledge to know about it. So I believe it's really an impactful period with Doctor. Thank you so much, ma. In fact, it's really a great privilege having you around, ma. It's awesome having you around. I
Thank you so
really, really
much, sir.
appreciate
Thank you so much,
your
man.
time.
Thank you
Thank
so
you
much,
for
sir.
your
Also,
time. Thank you for
Thank you,
everything, for
everybody.
your time. In fact, it's really, really a great time with you, man. Thank you
Oh,
so much,
thank
man.
you, sir.
Thank you so
Thank you
much.
for the invite. I'm very grateful.
It's an honor, man. Thank you so much, man. Thank you so much for coming on the station, man. And to round it up, as I said earlier, on, we are open to recruitment. We are recruiting membership in terms of probably you want to volunteer for any of our workshop activities, or you want to be a member, a full-time member. The slot is very much open to you. So you can always reach out to any of the leader of the group. Mr. Shegun is there, and so many other. Mr. David is there as well. You can also come into my DM as well. So we are very much open to recruiting and every other. So at this note, I believe we are coming to the end of the meeting of today's webinar. It's a great one, man. Thank you so much once again, man. We really, really appreciate you. Thank you so much, man. We appreciate you.
Thank you,
Thank you,
everyone. I'm very
everyone.
grateful.
Thank you very much, ma.
Thank you.
Thank you.
So,
Bye.
everybody, have a nice evening. Bye.
Have a good